Today we will discuss about the polypeptides and their synthesis. What are polypeptides? As we know that amino acids are bifunctional molecule with amino group at one end and carboxylic group at other end. These two functional groups when condensed with each other and forms the amide linkage by the elimination of water. Such type of linkage is also known as peptide linkage, and the product is known as polypeptide, peptide, dipeptide, tripeptide, etc. And how it is formed? As I have said that when two or more molecules condensed with each other, one amino acid and second amino acid molecule. When they condense, one water molecule will be eliminated and then they form a peptide linkage. This is known as peptide bond, and this is known as peptide linkage. And the product is known as peptide. This linkage is also known as amide linkage. Same as these two molecules condense, more than 2, 3, 5, when condensed, there is so on, then the name of the product is polypeptide. The resulting molecule contain a free amino group as well as carboxylic group. This is the free amino group and this is free carboxylic group. At one end amino group, at another end there is a free molecule of carboxylic group. These group may also react with other molecules of amino acid to form a high molecular weight or linear or condensed product. The formula of all the resulting condensation product indicate that every two amino acids are linked by means of CONH. This CONH linkage, which is known as amide linkage, peptide linkage, and the bond between of these two molecules is known as peptide bond. It is very important to note that a peptide formation between two amino acid molecules may react in one or two methods. For example, if here is glycine amino acid, 1H, or may be written as CH2, and here is the amino acid alanine, there will be attached a CH3 molecule. This product form is known glycyl alanine, because here is glycine and here is alanine amino acid. When they condense, the name will be glycyl alanine. If there is alanine molecule of amino acid is first, then the reaction will be, here is alanine, when it condensed with glycine, this is glycine, then one molecule of water will be come out and the product will be the name will be alanyl glycine. Same as when more than these two molecules, if one or amino acid is attached, same or differ, the name will be describe the product by the amino acid's name. If there is also alanine, glycine, and when alanyl glycine is formed and again one molecule of glycine is attached, then again one water molecule will be come out and the product will be named as alanyl glycine, again glycine, because there is two molecule of glycine are here, and this is a tripeptide molecule. Hence we can say that the formation of peptide is take by two ways, in which one I have told you, firstly alanine molecule is attached, then glycine molecule is attached. If glycine is first, then the name will be glycyl alanine. If alanine amino acid is first, then the name will be alanyl glycine. And if again one more molecule of amino acid is attached to form the tripeptide, then the name of the amino acid is also attached after the name of dipeptide, that is alanyl glycine, and if attached glycine, then alanyl glycine and glycine. If there is another amino acid by the name is can be alanine, again the name will be alanine glycine alanine. So there is two ways by which we can choose the preparation of polypeptides. Now comes on the synthesis of polypeptide. How there are different methods and principles by which we can say the polypeptides can be synthesized. First we will discuss about the general principles of polypeptide synthesis. For the synthesis of polypeptides, there are mainly 4 points constitutes the basic principles of all the methods. First will be protection of amino group. As we know that there are two free groups, which are one end is amino group and other end is carboxylic group. So first principle is the protection of amino group. In the synthesis of polypeptide, the first step is to protect the amino group. The protection of amino group is essential to avoid the interaction of two molecules of the same amino acid. The main protecting group are phthalate and carbobenzyl oxy group. Now comes on the reaction. How will we show the reaction of this group? This is phthalate. When this is attached with one amino acid, then the both hydrogen of amino group condense with this oxygen of phthalate and water molecule will come out. And the reaction will give the product is this. If this is the phthalate group, if we react with carbobenzyl oxy group, then the reaction in it the ethyl alcohol will be come out and the product will form this. This is carbobenzyl oxy group. Now second one will be the activation of carboxylic acid. First principle was protection of amino group. Now second one is activation of carboxylic group. The carboxylic group of amino group protected amino acid is activated by converting it into acid chlorides or esters or mixed anhydrides, and the reaction will be This is the amino group, protected amino acid. Where X is the protected group, which is attached at amino group, and the product will be acid anhydride. There is the activation of carboxylic acid group and then forms acid anhydride. As we know that the nature of carboxylic group is acidic. Now third step is coupling step. It is actual step where the peptide bond formation takes place. The formation of peptide bond is generally achieved by the nucleophilic attack of the free amino group of an amino acid ester or peptide ester on the activated carboxyl end of the N-protected amino acid, and the reaction will be this is the activated carboxyl compound. When it reacts with another nucleophilic amino compound, this is the nucleophilic amino compound. Then the product will be, there is a peptide bond formation in between this CONH, this CO and this NH. There is a peptide bond. And the product is named as dipeptide. Again here is X is the protecting group, which is protected the amino group of amino acid molecule. Now next is the step is removal of protecting group. After the formation of a polypeptide chain of desired length, when the desired length polypeptide chain is formed, then it is necessary to remove the protecting group. The protecting group is removed to form the final product. For the removal of different types of protecting group, various groups are used. I will show you two types of reaction by which the protecting group has removed. So on means the length will be whatever molecules are present in this polypeptide chain. When it's react with hydrazine, the formula is NH2NH2, this is known as hydrazine. Then this hydrazine is reacts here, one NH goes with CO group and one H goes with nitrogen, and the product will be this. Here these nitrogen and hydrogen of the hydrazine is react with this carbon and second one is react with this, and the product, the final product, this is the polypeptide chain. the final product we have got. This is first type of method, and now second type of method. When it goes with hydrolysis, then OH goes with CO and H goes with NH. The final product is polypeptide chain, and this protecting group comes out. This is the final product, the polypeptide chain, which we want. Now, second type of synthesis method is general method for polypeptide synthesis. The synthetic study of polypeptide was introduced by Fischer. After that, several other methods are introduced for the preparation of polypeptide, which are as first one is Fischer's second method. In this, Fischer proposed a polypeptide by the condensation of an amino acid whose amino group is protected by means of ethyl chloroformate. And the reaction will be C2H5OCOCl. This is the formula of ethyl chloroformate. When it reacts with one amino acid, then this Cl and this hydrogen comes out, and the formation is of C2H5O, CO, NH, CS2, COH. When it reacts with C2H5OH, that is ethyl alcohol, then C2H5O, CO, NH, CS2, COO, C2H5. When it reacts with another molecule of amino acid, there can be any type of amino acid, acidic, basic or aromatic, then ethyl alcohol comes out, C2H5OH comes out, and the product will be C2H5O, CO, NH, CS2, CO, another peptide bond is formed, CHR, COOH. Now the reaction goes under reduction, and when reduction forms, there is a 1H goes with CO and 1H goes with NH. Then the C2H5 COOH comes out, plus H and H, CS2, CO, NH, CHR and COH. The peptide. This is the peptide formation. This is a Fischer's second method. Now comes on the Fischer's third method. Fischer's third method. This method is actually the modification of above method, because in Fischer's second method, the protecting group is not easily removed. Hence, in this method, para-toluene sulfonyl chloride is used as a protecting group, which is easily removed by the means of hydrogen iodide, and the reaction will be CS3 SO2Cl. This is para-toluene sulfonyl chloride. it reacts with ethyl ester of glycine. This is the ethyl ester of glycine. Then this Cl and this H is condensed and formation of HCl, and they come out. Is SO2 NH CH2 COO C2H5. When it reacts with another ethyl ester H NH CH COO C2H5 and attached is one methyl group. This is the ethyl ester of alanine. Now reaction is continued here. CS3 benzene ring, that means this is a para-toluene. SO2 NH CH2 CO NH CH CS3 COO C2 H5. Now hydrogen iodide, which goes under hydrolysis at 50 to 60 degrees centigrade. Then ethyl alcohol comes out, and the formation of thiophene. This is thiophene, thiophenol, and one dipeptide, glycyl alanine. This is glycyl alanine as formed by the Fischer third method.